Alright, so I need to correct something here because last time I said this was a co-repressor. And this is actually an inducer. So I just went back to the negative inducible. Here we have an active repressor, and the substrate comes on and induces transcription. So it's still an inducer. So basically, whenever you have inducible operon where transcription is normally off and is being turned on, our little guy here that's helping, what I'm calling a co-regulator, I don't know if anybody else calls it that, but let's just say it's a co-regulator. Under an inducible operon, that co-regulator is an inducer. Whereas under a repressible operon, that co-regulator is a co-repressor. So here in our last one that we have negative repressible operon, we have our repressor. Normally we have transcription on, our repressor is off, and our co-regulator in this case is still a co-repressor, which assists the repressor in binding to the operator. So here, inactivated by co-repressor. Okay, those are operons. This slide just kind of gives you an overview of each type for review. Okay, clicker question. I'll give you a moment, and then we'll go over the answer. So they're asking, in a negative repressible operon, the regulator protein is synthesized as a. So they want to know when it's first synthesized, when it's first translated, is the protein that comes out, is it active, is it inactive, is it an activator, and is it a repressor? So in a negative operon, we have to have a repressor, so we know that it's either C or D. In a repressible operon, we know that it is automatically on, it needs to be turned off. So the answer needs to be inactive repressor, because if it's a repressor and the operon is normally transcribing, then it can't be active. It needs to be inactive. So the answer is D. Okay, another one. So for a blank operon, transcription normally does not take place and it is kept off by a blank. So if transcription normally does not take place, that makes it an inducible operon, and if it is kept off, that implies that it is maintained in an off position, so it needs to be a repressor that's doing that. So the answer is B. All right, so let's move on to, so we've been talking about operons in abstract, random examples. So let's talk about the very first and most famous operon, the Lac operon. It was first discovered in 1961 by Francois Jacob and Jacques Monod, and in 1965 they won the Nobel Prize for Physiology or Medicine for this discovery. And the reason why it was so significant is for two reasons. First, it gave us an idea for the first time of how genes are organized in bacteria. We didn't know this. And second, it elucidated our understanding of how these genes are regulated. And it's actually pretty elegant, I think. Okay, so a little bit about the Lac operon. Basically, it mediates the pathway for lactose digestion. So lactose is in milk, and in mammals who make and drink milk, lactose is in our guts and help us digest, or rather, E. coli that are in mammalian guts have the ability to digest lactose for us. So this is accomplished by genes in the Lac operon, and they encode beta-galactosidase, which is the main enzyme that actually breaks down lactose, and it breaks it down into galactose and glucose. Beta-galactosidase can also break lactose down into allolactose, which can then be broken down again by beta-galactosidase into galactose and glucose. And so beta-galactosidase can convert lactose directly to galactose and glucose, or it can do it via allolactose. The lac operon also encodes a gene that makes the permease protein, which is a membrane transporter protein that helps get lactose into the cell. All right, so what does the lac operon look like? So basically we have our lac Z that encodes the beta-galactosidase, lac Y encodes our permease, lac A encodes a transacetylase, which we're not going to talk about. Again, we have our lac O operator, our lac P, which is our promoter, where the RNA polymerase binds. Now, somewhere else on the bacterial chromosome, there's another gene that encodes the regulator for for the lac operon, and this regulator is called lac I. So normally the lac operon is off. That makes it what? Inducible or repressible. If it's off, it needs to be inducible. So it's inducible, and it is regulated by a repressor, which makes it negative. So this is a negative inducible operon. And the regulator gene also has its own promoter, PI. So that's pretty much the whole of the lac operon. So how this works is when transcription is on, the operon is transcribing beta-galactosidase, permease, and transacetylase. And this pathway is converting, all of these together are converting lactose into glucose and galactose. So what happens is that so normally the repressor is active, and it's normally on the operator. When allolactose is present, it binds to the repressor and basically removes it from the operator. So what this means is that the product of lactose metabolism, allolactose, is actually used to induce transcription of the lac operon. This is a little weird because it means that the end product, what you might think of as an end product of the pathway, is actually activating the transcription, which normally it's the substrate, right? The answer to this enigma is that we get some transcription of the lac operon all the time, but it isn't until the repressor is removed that we get high levels of transcription. And so we get there's some production of permease happening such that, and some production of beta-galactosidase such that when lactose does become available, permease can bring it into the cell, and beta-galactosidase can start making some allolactose. And once allolactose is present, then it turns on. It helps to induce expression of the lac operon. So, in the presence of allolactose, the lac repressor, and the answer is C, cannot bind to the operator. Okay, so that was the lac operon. And so normally E. coli, they prefer to use glucose because glucose is easiest for it to metabolize. So glucose is their preferred food source. But if glucose is absent, then they can upregulate alternative pathways that let them metabolize things like lactose and other other metabolites. So how is this? So this is basically called catabolite repression, where normally there's repression of expression of other sort of non-glucose metabolic pathways. But then when glucose is absent, they are stimulated. So the way this works is through positive control. And so basically meaning that there are activators involved and in other pathways. And that's a little weird because I just said that the lac operon is a negative inducible operon, but it's also controlled through positive control, positive regulation. And this is because it has a repressor, but it also has an activator. And this activator is called the CAP, or the catabolite activator protein. And so operons can be both negatively and positively controlled simultaneously. And so this catabolite activator protein is generally where it sits is slightly upstream of the lac promoter, and basically it requires this molecule. It's called adenosine 3'5' cyclic monophosphate. We call it cyclic AMP. Cyclic AMP is necessary for the CAP protein to actually bind to operons and upregulate them. And the availability of cyclic AMP depends on glucose availability. So if glucose is low, CAMP availability, or CAMP production gets stimulated. And so then it can induce binding of this CAP to a bunch of other sites to stimulate production of alternative pathways to glucose metabolism. So again, when glucose levels are low, CAMP production is stimulated, promoting binding of the catabolite activator protein to a number of different operons. So the CAP actually controls about 20 or more other operons that are responsible for alternative metabolic pathways to glucose. And so likewise, when glucose is high, CAMP levels drop, and these alternative pathways get shut down. So this is just the idea. Two take-home messages. CAMP is directly tied to glucose production and induces the activation of alternative pathways to glucose metabolism, and that operons can be both positively and negatively regulated at the same time. So this is just what this protein looks like. Here's the DNA strand that it's interacting with, and it's interacting in the major grooves of this. And here's the CAMP that changes it to the proper conformation so that it can do its thing. Quick clicker question: what is the effect of glucose on the lac operon? So glucose basically decreases transcription, so little transcription takes place. Okay, couple other things super quick. In some mRNAs in bacteria, they contain regulatory sequences called ribose switches, and and these bind to, they form secondary structures, and they bind to other proteins or molecules to induce changes that prevent translation from happening. Also, you can get RNA-mediated repression, where parts of the RNA can act as a ribozyme, and such that it interacts with mRNA, basically, with a protein, and induces cleavage in its own self-destruction. So this is regulation past transcription.